Okay, so now we have the Krebs cycle, which again is happening in the mitochondrial matrix. And we have produced acetyl-CoA, and my writing's gone dreadful. So we've produced 2 lots of acetyl coenzyme A, and the acetyl group has got 2 carbons in it. Okay, and then we start the citric acid cycle, or the Krebs cycle, or the citric acid cycle, or the TCA cycle, whatever you want to call it. So the first thing we do is we add that acetyl-CoA, or the acetyl group, onto oxaloacetate. Now the oxaloacetate has got 4 carbons in it. Okay, so when we add 4 carbons to 2 carbons, we make a 6 carbon compound, which is citric acid or citrate. So citric acid, hence the citric acid cycle, and that's 6 carbons. Okay, now because we've got 2 lots of acetyl-CoA coming through, we obviously need twice as much oxaloacetate, and we make twice as much citric acid. Now when the acetyl-CoA binds with the oxaloacetate, the CoA then is recycled. So this CoA then can go back into the link reaction, ready to pick up another acetyl group. So an important part of this step, not only is it taking the acetyl group into the TCA cycle, but it's also allowing coenzyme A to be reused. Okay, so then we have citric acid, and then we go to, and this is very simplified compared to what you've got, but we've got alpha ketoglutarate down here. This is 5 carbons, and we've got 2 of them because we've got 2 lots of citric acid. And then because it's a cycle, we have to have a regeneration point, so that we've always got some oxaloacetate to grab that acetyl group from the acetyl-CoA here. So we need to regenerate that by using some of that alpha ketoglutarate to go back to oxaloacetate. And in that bit of the reaction here, you've got the malate, the succinate, the fumarate. You've got a lot more detail on your TCA cycle than there will be here. Alright, so along this TCA cycle, you've got this metabolic hub, and you've got lots of electrons coming off. So at this point here, you've got some reduced NAD being made. 2 lots of reduced NAD being made there. Over here, you've got 2 lots of reduced NAD being made. And you've got 2 lots of FAD being made. So flavin adenine dinucleotide. Just these are electron carriers, just picking up the electrons that are being taken off by dehydrogenase reactions, and you can't lose that energy in the electrons or in the hydrogen atoms, so you need something to pick up those hydrogens, and it's FAD and NAD. There's also another 2 NAD coming off over here as well, because you've got a lot more steps obviously than I've drawn on here, a lot more opportunities for these dehydrogenation reactions. You've also got opportunity here to make some ATP by substrate level phosphorylation, so just having enough free energy when you've got a reaction happening here for ADP to be phosphorylated. So you can make ATP there by substrate level phosphorylation, which is what we did at the end stages of glycolysis as well, phosphorylation. So that's when you've just got enough free energy to take some phosphate and add it onto ADP. Alright, substrate level phosphorylation. So you've reduced a lot of electron carriers. You've given them these high energy electrons for them to carry into the next stage, the electron transfer chain. You've also taken out some carbon, because going from a 6 carbon to 5 carbon, you're going to lose some carbon dioxide here. Again, that oxygen doesn't come from your breathed in oxygen, it comes from the citric acid. And you've gone from 5 carbons here to 4 carbons here, so there'll be a bit of carbon dioxide coming out here as well. And that's where most of your waste carbon dioxide that you breathe out comes from, this TCA cycle. So you produce a little bit of ATP, so your products of this reaction, 2 ATP, a little bit of ATP. You've got 6 lots of reduced NADH that are carrying those high energy electrons, but you've also got 2 lots of FAD, reduced FAD. Okay, so you've got 2 different electron carrying molecules, NAD and FAD, and they will take those high energy electrons forward into the electron transport chain, and that will drive ATP synthesis. So obviously you've got lots more stages on yours, because you, like I say, you've got the succinate and fumarate bit on this side, on the left hand side. But it didn't really go into a lot of detail about enzymes and things, I don't think, from memory. But anyway, that was the basic. Alright, so basically you're stripping off hydrogens. It is dehydrogenation reactions all the time, stripping off those hydrogens which contain those high energy electrons, and then catching them using these electron carriers, and then these electron carriers will release the electrons down the electron transport chain, and that will be coupled with ATP synthesis. Alright, and there's a little bit of CO2 being given off in the process too. Alright, and a wee bit of just free energy. Alright, I hope that makes sense. Hello again. So let's have a look at the link reaction now. So we've made pyruvate in the cytoplasm, and that has a lot of potential energy in it. So we need to further break that down and oxidize it, so that we can release more of that potential energy and more of those high energy electrons to produce ATP. So this is done in the link reaction. So the pyruvate moves from the cytoplasm into now the mitochondrial matrix. Okay, so it's obviously permeable to that pyruvate, although some textbooks did say that the pyruvate goes into that mitochondrial matrix via active transport. So that means that it's going against concentration gradient, so I'm never quite sure about that. It will also only do this if oxygen is available. So only if oxygen is available. If oxygen is not available, then we get anaerobic respiration and no link reaction. Alright, so this was very easy at A level. It looks like it's got a little bit more complicated at degree level. So we've got 2 lots of pyruvate that have got 3 carbons, and they are then changed to acetyl coenzyme A. It's not a one-step reaction because it's not that easy. Acetyl CoA. Actually, just thinking about what I said then, I think it is a one-step reaction, but we need to have a CoA here. So CoA is this big protein complex. It's got thousands and thousands of carbons in it, and the pyruvate has only got 3 carbons. So you change the pyruvate to an acetyl group, and then you add the carrier molecule of the coenzyme A. And when you do that, you have another dehydrogenation reaction. So you've got a dehydrogenase enzyme working here, dehydrogenase, and that's your pyruvate dehydrogenase, isn't it? PDH. Okay, which has allosteric, again, allosteric control of it, the inhibitors, so anything that's going to stop it working, will be acetyl-CoA. So when we've got a lot of the product, you get end product inhibition. So a lot of this acetyl-CoA will mean that that enzyme will stop working. If you've got plenty of NADH, so you've got a lot of reduced electron carriers that are going to produce a lot of ATP, you don't need to do this link reaction very much. If you've already got loads of ATP in the system, fantastic, you don't need any more link reaction going on. And fatty acids, there's a really good Khan Academy video on this part, and she mentions about fatty acids being used as a way of forming lots of acetyl-CoA, and that's from beta oxidation. And she mentions that if you've got a lot of fatty acids in the system, then you don't need to do this link reaction because you can go straight from fatty acids to acetyl-CoA. Promoters of this enzyme, therefore, would be if we've got an abundance of oxidized NAD that needs to be reduced, if we've got too much CoA sloshing about the system, if we've got loads of pyruvate, that's going to drive the reaction forward because we've got a high concentration of reactants, AMP, adenosine monophosphate, and calcium ions. Because if we've got lots of calcium ions in our system, we need calcium ions for muscle contraction, and muscle contraction obviously uses ATP to move the myosin head, is it? So we need lots of ATP if there's lots of calcium. So the dehydrogenase enzyme will take off the hydrogen, and in most cases it will reduce our oxidized NAD to NADH and proton. Okay, but because we've got two pyruvates, we need two of those, we get two of those, and we get two of those. Also in this step, we're going from pyruvate, which is 2 carbons, to acetyl-CoA, pyruvate's 3, acetyl-CoA is 2 carbons. So we're taking off a carbon molecule, atom, and adding oxygen to it. But the oxygen actually comes from the pyruvate. It doesn't come from the oxygen you've breathed in, and that's a decarboxylation. So it's a simple step, the link reaction, but it looks like in your notes you have a lot of control over that. But it's very simple because it's all linked to your enzyme work and allosteric non-competitive inhibition. So that should be all right. Alright, so the link reaction happens in the mitochondrial matrix only if oxygen is available and pyruvate gets converted to acetyl coenzyme A, which is going to then start the Krebs cycle. Okay. Okay, Matty, let's go again then, see if we can sort this out. So we'll start with glycolysis. So this is occurring in the cytoplasm of the cell. Okay, and we're starting with glucose as our hexose sugar. So it's got 6 carbons. Okay, the first thing that occurs here is that we get a phosphorylation happening. So glucose becomes glucose phosphate, but it still has 6 carbons in it. And this phosphorylation increases the energy of the molecule, and it also means that you're increasing the negative charge, so it keeps the glucose actually in the cell. Okay, and you've been told now that the enzyme is a hexokinase enzyme, and again that will have allosteric regulation. So if there's lots of glucose in the system, then that's going to activate the hexokinase, and if there's lots of glucose phosphate in the system, that's going to be what we called end product inhibition. So you get end product inhibition here, end product inhibition, to prevent that enzyme working. Alright, so from glucose phosphate, we then do an isomerization to fructose phosphate, and it gives you, I think in the anaerobic notes, it gives you all of the details of that. But basically, you just need to use an isomerase. Okay, and then we need to phosphorylate this again to make it fructose biphosphate. And you probably need to know the enzyme there. Let me get my notes that I took earlier. It's phosphofructokinase. Okay, so phosphofructokinase. So again, you need to just refresh yourself about allosteric activators and inhibitors here. So the inhibitors, which are the minus ones here, are ATP. So if you've got enough ATP in the system, you don't need to make any more. So that's going to stop this enzyme working. And citrate, which is the first product of the citric acid cycle. So if you've got enough of those, then you don't need this reaction to happen. But if you've got lots of AMP in the system, then that will activate this enzyme. And if you've got lots of fructose biphosphate, then you will also activate the system and drive it forward. Okay, now you've got this 6-carbon phosphorylated sugar, biphosphorylated sugar. You can then split it, so you're doing the splitting, the lysis of glycolysis here. And you've got glyceraldehyde phosphate, okay, which is 3-carbon because it's half of 6. And you've got the other isomer, which is dihydroxy acetone phosphate, which presumably is allowed into the mitochondria through the first membrane anyway. But then you have this isomerase at A level you did, isomerase, to turn it into that dihydroxyacetone into glyceraldehyde. So now from here on in, we go everything twice. And I'm hoping that as I move this screen up that it gets captured on the video. So let's have a look at what happens to glyceraldehyde phosphate. Well, we need some inorganic phosphate coming in here. So it's not coming from ATP. Oh, and I've forgotten that. Dearie. Okay, so the phosphate here to add onto the glucose comes from ATP here, which is daft because we're supposed to be using respiration 4 to make ATP, but actually we're using some up. And again here to phosphorylate your fructose phosphate, we need some ATP. So we've already used 2 ATP. Alright, but at this point here, you don't use ATP to add the phosphates. You just use inorganic phosphate. And you make glycerate biphosphate, or bisphosphate, or diphosphate. The bis, bi, or the di. What is it? It's the carbon that you're attaching the phosphate group that determines whether it's biphosphate or diphosphate, but this is glycerate biphosphate. Now when you change glyceraldehyde phosphate to glycerate biphosphate, as well as phosphorylating, so this is phosphorylating when you're adding phosphate, you will also dehydrogenate your glyceraldehyde phosphate. So you'll have dehydrogenase enzymes working here, stripping off hydrogen from the glyceraldehyde phosphate. So dehydrogenase enzymes will be working to remove the hydrogen. And the molecule that they put them onto is an NAD molecule. So you'll have oxidized NAD in the cytoplasm. You'll allow it to pick up the 2 hydrogens that are stripped off glyceraldehyde phosphate, and that produces NADH plus a proton. And because you're doing it twice, okay, it's 2 of that, actually 4 of that, okay, and 2 of that. Alright, So let's move on then to what happens to glycerate biphosphate. So now we can start getting back our ATP. So glycerate biphosphate just becomes glycerate phosphate, and we need to have 2 in front of that and a 2 in front of that. And then what comes off here is obviously phosphate, and we can use it to add it to ATP to make 2 ATP. So we've already recouped the ATP we used earlier. And then finally we go to pyruvate. That's our final end product for glycolysis. And again, we're taking off that phosphate so we can phosphorylate 2 lots of ATP into 2 ATP. So what we have is 4 ATP that we've made minus the 2 ATP that we used above. So that's a net of 2 ATP. But we've also got 2 lots of reduced NAD, which will then have to get transported by the more involved mechanisms that you're learning about into the matrix. So it won't be in this form, but something will happen to take those hydrogens off there and take those through into the matrix so they can be used to create ATP. And you've also got 2 pyruvate that can go into your link reaction. Okay, so I'm going to stop there and see if that works.